And let's see. All right. So I would like to welcome you all to our Firefly Watch training this evening. We're so happy to have you join us for our program. We would like to welcome our old Mass Audubon friends and also welcome new Mass Audubon friends. We have folks joining us from across the country this evening, which is super exciting. And we're so excited that you guys are all looking forward to learning more about fireflies with us. For those who are not familiar with our organization, with more than 125,000 members, we work to both protect and steward over 38,000 acres and make nature accessible to all in Massachusetts and beyond through our advocacy, education, and conservation programs. As I mentioned, we are super excited to have you all join us in this mission by taking part in our Firefly Watch Citizen Science Project, starting with this training tonight. My name is Robin, and I am the School and Public Programs Director at our Drumlin Farm Wildlife Sanctuary, and will also be your moderator this evening. So just a quick few housekeeping things just to get everybody oriented before we get started. First of all, I'm just going to switch the screen that I am sharing so that I can orient everyone. What I would like to do is just ask folks to turn off your cameras if you are not one of our speakers, and to keep your microphones muted so that we can make sure that we can focus and keep our bandwidth good. And then what I'm going to do is I'm just going to share my screen so that I can just sort of orient you all for folks who may not be familiar with Teams. And so what we will do, so if you guys can see my screen right now, and you can see my, my cursor here. At the bottom of your screen, if you just gently toggle over the bottom menu little here, it'll pop up a bar. And that bar is where you can control your camera, your microphone, and as well as a few other things. So if you click on the three dots, the more actions, you can jump into full screen. And then you can also turn on live captions if you want to have those sort of playing in the bottom left-hand corner, which can be helpful sometimes as we're moving through. The other piece is this box right here is our chat box. And so if you click on that, you can have that open. And if you look down here, this is where you'll enter your chat. And then, and what we're going to use chat for this evening is actually for asking questions. How we're going to run this session this evening is we have two speakers joining us, and we have so many people with us this evening that we're going to sort of do probably about every 10 or 15 minutes question breaks. So if you guys have questions, please, please, please type them in chat as they come up for you. And that way I can keep track of them and we'll make sure to ask them when we take those question breaks. We want to make sure that we get everyone as much information as possible this evening and answer as many questions as possible this evening. So with that being said, that pretty much orients you to our screen. There's not a whole lot else that you need to know. And so what I'm going to do is I'm actually going to go back to sharing my other screen so that I can introduce our speakers. So if you guys give me 1 second so I can flip over. And then, all right. So, and then do that. Okay, and so then what we will do is, all right, so what I would like to do is actually introduce our speakers for this evening. And so our first speaker that we have is Chris Kratsley, and he is a professor of biology at Fitchburg State University, where he teaches about behavioral ecology, scientific research methods, and the intersection between science and society. Dr. Kratsley's research program focuses on the courtship, mating, and predator avoidance behavior of fireflies, with a particular emphasis on the evolution of bioluminescent signaling behaviors. His current, oops, sorry, just dropped that. His current interests involve exploring the evolution of chemical defenses in fireflies and partnering with Mass Audubon to foster citizen science efforts through Firefly Watch and Firefly Watch Pro. And then our second speaker is Clyde Sorensen, and he is an Alumni Association Distinguished Undergraduate Professor of Entomology at North Carolina State University, where he teaches about the interactions between insects and people, arthropod pest management, and insect natural history. He conducts research on the reproductive ecology of rare and endangered plants of longleaf pine savannas, insect pest management, and the natural history of insects, including ants, Lepidoptera, and others. He also has a deep interest in the fireflies of North Carolina and is working to understand the diversity and distribution of these insects in the state. So again, welcome and thank you for choosing to spend your evening with us. Just to give you an idea of what our evening is going to look like so the training is outlined, as you can see on the slide in front of you. And so we're going to start with Clyde Sorensen doing his femme fatales and mountain ghosts, fireflies and their relatives, which is an overview of North American firefly biology with a little North Carolina flavor. It's a good time to have that, I would say. And then we will move on to Chris Kratzley, who will be talking about his monitoring firefly populations as part of Firefly Watch, and an introduction to identifying fireflies in the field with a little Mass flavor. So we're getting a little Southern and a little local. Well, local for us, not so local for a lot of folks who are joining us this evening, but we'll try to cover as much as we can. And then identifying firefly genre and flash patterns, monitoring and entering data on firefly populations through Firefly Watch, which is the platform that we use to track all this data, and monitoring, entering data on firefly populations through Firefly Watch Pro. So on that note, I will hand it over to Clyde. All right.
How's everybody doing? I hope everybody's well. I'm going to go ahead and switch over to my PowerPoint presentation, and we'll go ahead and get started talking about firefly biology, if I can make all of this work. So sorry, folks.
No worries. And just a reminder to everyone, as if you have questions as Clyde's going through his presentation, please put them in the chat and we will take care of them as we move through.
All right. So what my goal is in the next little bit is to try and basically give you a good introduction to what fireflies are and some of the interesting aspects of their biology, and a little bit of information on how to go about identifying some of them. And I think Chris is going to be revisiting some of that as well. And so I've titled this Femme Fatales and Mountain Ghosts for a good reason. And I hope some of you already know what that's about, but I hope most of you don't. And we'll get to that a little bit later as to what the significance of those two terms might be. So what is a firefly? So it's important to know, first off, that fireflies aren't flies and lightning bugs aren't bugs. What they are beetles. And if you look at that picture over here, will my pointer work? Is that working? Can you all see my wandering cursor?
Yep, you are all good.
Yes.
Super. So this is one of the hallmarks of an animal that makes it a beetle, and that's the weird front wings that we call elytra. And so fireflies are beetles, and they belong to the family Lampyridae, and Lampyridae means light bearer. And you know, we as a species have been paying attention to them for a really, really long time. The ancient Greeks had a term for them. They called them Chrysolanthus, which translates basically to the one with the glowing butt. The Lampyrids are closely related to several other groups of beetles, including click beetles and some others, and a really cool kind of small family called Phagotidae, which are the true glowworms. And we'll talk a little bit about them later on, mostly because they're just really cool. It's important to note that there are other bioluminescent insects beyond fireflies and glowworms, and we'll talk a little bit about some of those a little bit later as well. But without a doubt, the most famous bioluminescent insects, and probably for most folks, the most familiar bioluminescent animals are fireflies. Lots of folks like to call them lightning bugs, but I'm going to be kind of haughty and call them fireflies. So this is just a quick diagram of the some of the significant parts of a firefly, at least from the dorsal side. And these are parts that are important for you to know if you're going to identify these insects, because these are the parts that frequently vary enough for us to tell one from another. So like all insects, they have a pair of prominent antennae. Those antennae can be different shaped in different species. They have a pair of compound eyes. And in male fireflies, the compound eyes can often be huge. If you think about what a male firefly's whole mission in life is to see a little bitty glowing dot somewhere below it. Having really huge eyes is a good thing. Behind the head on a firefly is a structure called the pronotum, and this is this structure right here, this shield-like structure that on most fireflies kind of protects the back of the insect's head. And as it turns out, the color, the shape, the markings on the pronotum can be really, really informative in helping you sort out just exactly which firefly it is you're looking at. Behind the pronotum is the remaining part of the thorax. So fireflies, like all other insects, have 3 pairs of legs, 1 pair per thoracic segment. The pronotum is associated with those front legs. And then behind that pronotum is a little tiny triangle, a structure called the scutellum. And that's another structure on a firefly that can help you sort out what you're looking at, because it could be colored one way or another. It's not going to vary in shape very much from one species to another, but it can be an important additional tool. Behind that are the elytra. And elytra is plural, elytron is singular. And on fireflies, the elytra, in most cases, if they have wings, and some fireflies don't have wings as adults, if they have wings, they cover pretty much the entire abdomen. And of course, they cover those big folded up membranous wings that we saw in the earlier picture. The maculations are simply patterns on the elytra, can be really, really important in helping you identify a firefly. In addition, on the elytra, on lots of fireflies, there's a little tiny crease that runs right here, and that's called the elytral fold. And that's yet another character that can be important in helping you sort out who it is you're looking at. Now, I don't have a picture of the underside. I have one later on that you'll probably notice. But on the underside, there are some important characteristics too that you might want to look for. One of them being just what is the color of the abdomen and the other underneath structures, and what are the shape and size and location of the light organs that the insect has that it uses to communicate with. And so there are, in addition to that, a couple other small characters that, if you get really into this, might become important. But these are the important parts of a firefly. All right, so just to give you a kind of a coarse idea of the diversity of fireflies in the world and in the United States. In the world, there are probably a couple thousand. And I said probably because, point of fact, nobody knows for sure how many species of fireflies there are. And without a doubt, there are hundreds, if not thousands, of undescribed species of fireflies yet to identify in the world. And there are some good reasons for that. We might get into that a little bit later. In the US, there are about 170 species or so. Again, that's probably an underestimate of the actual diversity of fireflies. And they're in 20 or so genera. So the genus is the taxonomic level immediately above the species. So a genus of insects is a group of insects that are very, very closely related and, of course, share a common ancestor. In North Carolina, we have somewhere in the neighborhood of at least 30 to 50 species. And just to give you an idea of how diverse even a small locality can be, in my yard, I so far documented about 10 species of fireflies on my little 4-acre property here in the central part of North Carolina. So they're not by any stretch of the imagination the most diverse beetles, but they are very diverse animals. In North America, there are 4 genera that are kind of prominent and conspicuous and frequently encountered. And I'll real briefly try and describe each one of those to you. The first one is Photinus. And Photinus is, I suspect, for most people, by far the most familiar. And this is probably, for most folks in Eastern North America, the most familiar Photinus. This is Photinus piralis. Some folks call it the Big Dipper. We'll talk about him a little bit later. Unfortunately, I understand you folks in Massachusetts don't have piralis, but you know, it's always a good idea to keep your eyes open. You never know what might be changing. So Photinus fireflies range from fairly small to relatively large. What's that mean? The largest ones are maybe a ½ inch long, and some of the smaller species are only about a ¼ inch long. They tend to be kind of flattened when you look at them from the side, and most of them, unfortunately, look very similar to Photinus piralis right here. So telling them apart by physical appearance characters can be kind of interesting. The next genus that you're most likely to encounter, one that is quite common and quite widespread, is Photurus. And this is Photurus versicolor. This is one of the more common ones. So Photurus fireflies tend to be big. They tend to have really prominent elytral folds, as I mentioned earlier. And lots of the species have kind of an arrow-shaped black mark on the middle of their pronotum. And one real defining characteristic of Photurus, well, a couple defining characteristics of Photurus is when you look at them from the side, they look kind of hunchbacked. They kind of bend where the pronotum and the elytra meet. And they also tend to have long legs compared to other fireflies. And there's an important reason for that we'll get into a little bit later. This is a diverse group. Some of the female Photurus have interesting behaviors we'll talk about a little bit later. And this is one that unfortunately is not real well known. There's no doubt several undescribed species in it. And unfortunately, some of these fireflies also have pretty plastic flashing behaviors, which means they might change what they're doing from one time to the next. And that can make things a little more confusing. Pyractomina is the third genus that you might well encounter in many parts of North America. These tend to be kind of large-ish fireflies. Photurus is big, by the way, too. Some Photurus are maybe approaching 3/4 of an inch long, but that's kind of on the outside. Pyractomina can be large as well. They have a kind of a defining shape to their pronotum. If you look at this individual right here, let me get my cursor back, you notice that the pronotum's kind of got a peak to it, and it covers more of the head oftentimes than you see on the other genera that I've talked about. And notably, a lot of the Pyractomina have these dark markings on the sides of their elytra. And some of these are really early spring flying species, and so they may be some of the first fireflies you would encounter in the spring. Some of these also have interesting flash colors. So most Photurus tend to flash kind of a green color. Lots of Photinus flash green. Photurus tend to flash either green to yellow. Some of the Pyractomina flash kind of an orange, or even to some people's mind, a red. The fourth genus is Phausis or Phausis, and these are the ghost fireflies. That's just an advertisement for a really great beer up in Asheville, North Carolina. This is actually what the fireflies look like. This is probably the most well-known species in this group. This is the blue ghost. And these are interesting because, for one thing, they're tiny. Most of them are much smaller than less than 1/4 of an inch long. And the females in this genus are wingless. So the picture that you're looking at over here shows a mating pair. This is the female and this is the male. And a lot of these in this genus don't flash so much as keep their light on and fly around. And so they make a pretty spectacular display when you can see them in large numbers. The picture on the right here is showing kind of a time-lapse exposure of blue ghosts. Now, even though they're called blue ghosts, their light is actually kind of green. If you get up close to them, you'll notice that it's not really green, or really blue, it's green. And that's due to an optical illusion called the Purkinje effect, but we'll not worry too much about that right now. Okay. An important thing to recognize when you talk about lampurity about the fireflies is that not all fireflies are lightning bugs, because about a quarter of the species of lampyrids are day flying and don't have light organs. And in these species, and they look pretty much like regular fireflies, what you'll notice is that on the top it looks like a firefly, and on the bottom there aren't any light-colored light organs. Some of these, the males are dark and the females glow, and many of these other life stages may still glow, and either I or Chris could address why that might be. And these fireflies that aren't lightning bugs appear primarily to rely on pheromones for communication. And here's a pretty notable example of one of these. This is Lucidota, and this is another one that I have in my yard here in North Carolina. So bear in mind that when I said 170 species in North America, a fair number of those don't make lights. That doesn't mean they're not important, but it does kind of complicate trying to find them. All right, so another important thing to know about fireflies is that a lot of insects like to mimic them. A lot of insects mimic fireflies because many fireflies are protected by noxious chemicals called lucibufficants. And these are chemically related to the toxins that we find in toads, which is an interesting observation in its own right. So there's a soldier beetle that looks a whole lot like a firefly. There's a longhorn beetle, a cerambycid wood borer, that looks a whole lot like a firefly. And there's a ground beetle that looks like a firefly. And it is important to recognize that some of these make their own nasty chemicals as well. And so they kind of reinforce the message that we're not good to eat by being noxious, just like the fireflies are. So all the beetles that you see that look like fireflies aren't necessarily fireflies. So what makes fireflies so cool? They produce light. They bioluminesce, and it is not just cool that they produce light, it's cool light that they produce. And the process is kind of a neat chemical reaction. The material, the chemical that is responsible for light production is luciferin. In order for luciferin to glow, you have to have oxygen in the reaction. You have to have an important enzyme called luciferase, and you have to have an energy source, and the energy source for all living cells is ATP, and then a couple other odds and ends. But the neat thing about this light production system is that basically all the energy that goes into the light production system comes out as light, and there's very, very little of that energy that enters the system wasted as heat. If you think about how we make light, everything we do to make light generates waste heat. Even LEDs generate some heat. But fireflies and other bioluminescent animals and other bioluminescent organisms have kind of outsmarted us. Of course, it took millions of years to get there, but that's a different story too. All right, just real quickly, this is one of the other bioluminescent insects that you might encounter in eastern North America. This is an adult female glowworm, It doesn't look like a beetle, but it's an adult beetle. And some folks have in the past called these things railroad worms. This is a picture I took of one that was coiled up on the bathroom floor in the upstairs bathroom at 2 o'clock in the morning. And I took this picture by taking a flash exposure on top of a time-lapse exposure. And what that does is it allows you to see where the light organs are on that animal. And these animals, the adult females and the larvae bioluminesce. Not real sure why they bioluminesce, apart from maybe it's a warning not to eat them. It may also have some role in how they prey. They feed primarily on non-insect invertebrates. They're pretty spectacular. One of the other cool things about them is, let me show you again. That's the female. That's one coiled up. That's the male. And they look nothing whatsoever like each other. Every time I find a male and a female, I'm just mystified as how they actually do the deed. But there you are, a really cool bioluminescent insect that's not a firefly. Another group of insects that I want to mention real quickly that are bioluminescent are some of the flies, the true flies. Probably the most famous of the true flies that are bioluminescent are the New Zealand glowworms, New Zealand and Australia. And these are the larvae of a fungus gnat that live in caves and grottos. And unlike fireflies, which use their flashes to signal within their species, primarily for mating, the New Zealand glowworms use their beautiful blue lights to help attract prey. And so they spin sticky webs. They hang from the ceiling of their cave with their little blue butts shining, and insects that wander into the cave fly up thinking they're flying into a starry sky, and what they're actually flying into are the jaws of death. Real interestingly, we have a bioluminescent fly in North America. It's found in the southern Appalachians, if not beyond. And according to at least one report, it's the bluest biological light known. So one of my missions this summer, if the COVID situation ever improves enough, is to go up and find some of these really cool bioluminescent fly larvae here in North Carolina. All right, so this is a good time for us to stop and take some questions. By the way, the picture that I've got on this slide here is giving you a size comparison between one of the smaller photinus fireflies. This is consanguinus, and a regular old Asian multicolored ladybird beetle. So if you have a house that's invaded by those ladybugs every fall, you have a good idea how big this firefly is. So with that, Chris and I will be glad to take any questions you might have up to this point.
So at the moment, we just have one question in the chat. So folks, if you do have questions that have come up for you, if you want to throw those in right now, that would be great. But to get us started, the question that we do have is, are there fewer species the further west you go toward the plains?
I think that's a pretty safe generalization, but like all generalizations, it's probably not completely accurate. But they're certainly far more abundant in the eastern half of the continent than they are in the west. And in fact, in many parts in the west, there simply are no fireflies. But you know, that this is still something that is not terribly well known across the continent yet either. So, but I think, you know, once you start getting into what was the prairie before European settlement, once you start getting into that part of the continent, yeah, fireflies start playing out pretty rapidly. Now, there are some species in the far west, but they're very patchily distributed, and I think several of them are in that we don't have a lamp category. You want to add to that, Chris?
Not too much, just that same final point you made, which is that many of the, you know, there are diurnal fireflies, the fireflies that are active during the day, you are perhaps more likely to find out west than the nocturnal ones, which seem to be the group that decrease in their species richness and their frequency as you head west.
Great, thank you. So along those same lines of where we can find our fireflies, does Florida have fireflies?
Florida absolutely has fireflies. In fact, it has some species that only occur in Florida. And a lot of the fireflies that are found in the southeast and in Florida in particular seem to be associated with various and sundry kinds of wetland edges. And fireflies in general like kind of humid environments. And that's probably one reason why they start playing out the further west you get. But Florida definitely has some unique species compared to the rest of the country.
Great. And then can you just repeat how many species are in Massachusetts again?
Chris will have to answer that.
Yeah, so one of the reasons that we began with Clyde talking from his certainly, North American, but also North Carolina perspective is you heard they have about 30 to 40 species in North Carolina. Massachusetts is more closer to 20 different species in Massachusetts. But as he's noted, as we've noted about fireflies across the country and certainly in our regions, it's always a question whether we might find another one. And it's always a question whether some that we saw, 50 years ago might be disappearing. And it's also, a question whether some might be expanding their range due to global warming. So we have to keep these numbers as ballpark figures for now, but I'd say that 20 is a good ballpark figure for Massachusetts.
Yeah, and to that point too, as I alluded to earlier, and I'm sure Chris would second, we still don't know a lot about a lot of these insects. They're kind of cryptic during the daytime, and there's behavioral plasticity, and there are other kind of weird things happening as well. For instance, we have what is almost certainly an undescribed species of ghost firefly here in the Piedmont of North Carolina. That means science doesn't know who it is yet, but we know it's there. We've just got to find enough of them to do some basic research on them. And this is a firefly that's obviously been there right along, but because it only flies for about 2 weeks, 3 weeks out of the year, and it only flies for about 20 minutes in the evening, and because it's only about a little over 1/8 of an inch long, it was a firefly that was very easy to overlook for a very long time. So there are a lot of really cool things like that still out there to discover. And that's one of the beauties of a program like Firefly Watch. It gives people an opportunity to make a discovery that might be new to science. And that's a really cool possibility.
That's very cool. Thank you. In just a general idea of what the lifespan of a firefly is?
Okay, so that's tricky because most of the life of a firefly is spent as a larva, living in leaf litter or in the upper parts of the soil, pretty much out of sight and out of mind. And so when you talk about lifespans, it could be a year, it could be 2 years, depending on some of the far northern species. It takes them a while to finish their development, and they do go all the way up into Canada, southern Canada. So when we talk about the lifespan, of the insect, it's a while, but the lifespan of an adult in most species is probably only a couple weeks. Weeks, 3 or 4 weeks.
Thank you. Can you speak to any behavioral differences between eastern and western species that do light at night?
So I really don't have a whole lot of insight into the western species, what few, if there of them, that might bioluminesce. I'm going to talk about just in just a moment some of the differences in species that occur around the eastern part. But behavior is really one of the most important ways we can identify or work on identifying fireflies, because unlike a lot of other insects, as I said earlier, in a lot of cases, there's not a lot of morphological, that means appearance difference between species. And a lot of the difference is how they behave. They're kind of what we call cryptic species in that respect. Chris, I don't know if you know much about the western blowers, what few there are.
Yeah, I don't have anything to add to that. I think it's a fascinating question. I guess I would only say that one thing, again, because of the pattern of greater diversity and frequency in the East, here in the East, you probably have an incredible range, or you do have an incredible range of different behaviors. And as you move to areas where you might, out west, where you might have only very isolated populations with only a few different species represented, certainly that diversity is going to narrow. And so I would say you probably have pretty much the full range of behaviors exist in eastern fireflies that you would see out west, but out west you would see just a few of those representative behaviors. I think that's probably the best way to describe the pattern.
I think that's correct. And one place out west that actually has some diversity of flashing fireflies is Arizona. Oddly enough, Arizona's desert. They don't like desert. Well, actually, Arizona is largely desert, but there are all these cool little oases. But in almost every place that there are fireflies in Arizona, there's only a single species that flashes.
Great, thank you. How about, do fireflies serve as pollinators?
Do y'all mind if I tell you a little short story?
We would love a short story.
About science and how sometimes when you learn is not what you expected to learn. One of my projects is on the Venus flytrap. We're studying the reproductive ecology of the Venus flytrap, which is arguably one of the most charismatic plants in the world. I certainly think so. It's got the wrong name, but that's a different story. Well, we're making collections of pollinators, of insects in the vicinity of fly traps, and we had the idea that we would put some time-lapse cameras with a red lamp so that most insects can't see red on the flower, and we would see if anybody came to the flowers at night. And so we put some cameras out and we put the little red lamps out, and we came back the next morning and we checked our video, and lo and behold, fireflies were landing on the Venus flytrap flowers. And we were thinking, oh, this is so cool. This is National Geographic stuff. We're going to get one of the most charismatic plants in the world pollinated by one of the most charismatic insects in the world. How cool could this be? So the next night we went out with our red headlamps on so we could collect fireflies off the flowers as they were landing on them, and they flew to our headlamps. So they weren't attracted to the flowers at all. They were attracted to the red reflecting off the flowers. As it turns out, fireflies can see red. But to get to your, and so, yeah, there goes the National Geographic story. But anyhow, to get to your point, fireflies may be pollinators to a limited extent, but they're probably not real important as pollinators. And if you look at them, they're not really built to be good pollinators because they really don't have a whole lot of like setae or hairs on their body to help trap pollen. Most good pollinators are hairy, and they collect pollen and then redistribute it. But I'm not going to say they're not pollinators, but they're probably not real important pollinators.
Great, thank you. Are all fireflies native or are some invasive?
As far as I know, they're all native. I don't know of any invasive species, and I'd be glad for Chris to correct me on that, but I don't think there have been any introduced. And, you know, if you look at the biology, and we're going to talk about that in just a second, but the biology of fireflies really doesn't lend them to being transported accidentally as readily as some other insects might be.
Great. And do they know why some fireflies glow and others don't?
That's a good question for Chris. I can take a shot at it, but it might have something to do evolutionarily with why they glow in the first place. And I think one of the main lines of thought right now is that they originally evolved this ability to glow not to communicate with others of their own species, but to warn potential predators that there were nasty things to eat. And we're talking primarily about the larvae in this case. And that evolutionary advance of light production was then later co-opted by some of them for sexual signaling. That's one theory. Got anything to add to that, Chris?
I think that that's a perfect description of it. And I would just add that we also think that many of these diurnal fireflies, the ones that don't flash, we think actually have secondarily lost the flash behavior. And Clyde, as he goes on with this lecture, will talk a little bit about one of the evolutionary pressures on fireflies from predation that might have driven some of them to to away from a nocturnal lifestyle. So.
Yeah, that's exactly right. So that's the thinking. And by the way, in a lot of those that don't have light organs as adults, a lot of those larvae still glow. So they still have light organs, and so they're still warning predators that they're nasty.
Right. Along the same lines of the glowing piece, how long do the more common fireflies light nightly?
Well, we're going to get to that in just a few minutes, but that's an important thing to know, and I think Chris is going to reiterate this. Different species of fireflies exhibit different flashing behaviors, and so some species start flashing while it's still pretty light outside, and they quit pretty much when it gets full dark. And some other species don't start until it's been full dark for an hour or more. So different species have different timing of their flashing behavior. And some species will flash for hours, and other species will only flash for 10 or 20 minutes every evening. So that's an important, actually, kind of thing to note, because when you're observing fireflies, because that can actually help lead you to an identification.
Great, thank you. And it looks like this might be our last question for the moment. Are fireflies more abundant in forested areas or open areas like meadows?
It depends. It depends on the species. So some species are very tightly associated with open habitats or edge habitats, and some other species are very much attached to particular, not just forest habitats, but particular forest habitats. And so the cool thing is, if you move around between habitats, you see more different kinds of fireflies.
All right, and it looks like a couple more just popped up. Hold on one second. Does weather and temperature have impact on firefly flashing activity?
Absolutely. If it's a driving rain, they probably won't be out. But it's pretty amazing how much rain they can tolerate. Most fireflies are not real active when it's really cool. Of course, some of those early spring fireflies, if they're not active when it's cool, they're not going to be active at all. So there's variation between species. But for a lot of the late spring, early summer kinds of fireflies, it needs to be over 55 or 60 for them to be very interested in doing very much. On the other end of the spectrum, if it gets really dry, a lot of times you'll notice that firefly activity goes down. And that may be because the adults don't want to come up and be desiccated, or it may have something to do with how rapidly they're coming up as adults. And so again, they tend to like humid environments, and when it's really dry, they tend not to be all that active.
Great. And I think that looks like it wraps up our questions for the moment. Thank you.
All right. So if y'all are ready, we'll go ahead, and I've only have a few more slides, but I do want to talk about what they're doing and how they do things. So get back to a little bit about firefly biology. Most of them are predators as larvae. They feed on other invertebrates, and sometimes the predatory relationships are phenomenal. So you may have little bitty firefly larvae killing fairly large slugs and snails, and feeding on them for quite a while. So, but they're pretty aggressive little leaf litter lions, if you want to put it that way. They do, as I said earlier, have a wide range of habitats: marshlands, grasslands, woodlands. Most are probably associated with, in one way or another, with woodlands or woodland edges. And for some species, it's really critical, particularly those species that have wingless female adults. It's really important that there's mature native forest that's been relatively undisturbed, because those species don't have very good dispersal abilities. Some exotic plants and invasive plants can be detrimental. We could get into that later, but I won't worry about it too much. Again, many of them are wingless and larviform as adult females. Most species, with a couple notable exceptions, don't feed as adults. In most fireflies, the main business of the adults is to simply successfully mate and make sure there's something going into the next generation. This, by the way, is what a firefly larva looks like. They're kind of neat looking. Most of them are bioluminescent, as we mentioned earlier. Okay, so what do they do with their lights? What's the main point of the lights on the adults anyway? It's communication with other members of their species, and sometimes with other species. And almost all of that communication is about sex. So fireflies, bioluminescent fireflies anyhow, have largely replaced what most insects do with pheromones with light. And as we said earlier, different genera often have different colored light. Each species has its own signaling system. And so what's that mean? That means in the typical case, the males, at least in most North American species, this is different in other parts of the world. In most North American species, the males fly around at the right time of night, in the right habitat, at the right height above the ground, making his species male flash signal. And that varies from one species to the next. While he's flying around, he's looking where he suspects females could be with those great big compound eyes for a female to respond. And most of the species we have, the females are either on the ground or in low vegetation, or sometimes up in the tops of trees, and they're watching for the males. And if she sees a male who's making the right signal for her species and doing a good job of it, that's a real important thing to recognize, then she'll respond with her species' female signal. And that, again, is typically in some way or another species specific. And if that male sees her signal and responds again, he'll start approaching her, flashing as he's going, and she'll be signaling back to him. And then eventually he'll get close enough to land near her. He'll run over there, they'll shake hands, and then they'll get down to business. And interestingly, in fireflies, they may couple for many, many hours. And they do that because in lots of firefly species, the male provides the female a really large, what's called a nuptial gift. which is a bunch of energy that she can use, bunch of energy and sometimes other chemicals that she can use to more successfully reproduce. So this is a real short little video I took in my yard that shows the consequence here. So what we have in this video is a pair of Phytinus paralis that have successfully gone through that whole process, shook hands, and now they're coupled. And there's a very frustrated additional male trying to horn in on the action. And if you look in the background, you'll see a third male flashing. So that poor guy's just out of luck, but he's not going to give up. And if you'll watch, you'll see a little in a moment. There he is. See that one in the background? So that gets to another interesting issue, and is all their communication light? It may not be all light. There may be other things going on as well. All right, so let me get out of here. This is the flash pattern for Phytinus paralis. This is the one I've been talking about fairly frequently here. You have to watch carefully. On that black screen, you'll see a green J shape coming at you. And that's the characteristic flash pattern for a male paralis. This is the big difference. There'll be another one. You see it? So he flashes about every, I think it's every 4 seconds. If a female sees him, she'll flash 2 seconds after he flashes. She'll flash for a half second. And if he sees that, then he'll go in that direction until they get close enough to decide they like each other. So that's paralis. That's its signaling pattern. This is another species of Phytinus called consanguinus. Some folks call it the double tap. And you have to watch this video very carefully because the light's not particularly bright. I took this in my backyard. But you'll see coming out of the left and going to the right a double flash. And then a short while later, another double flash. And then a short while later, another double flash. So there's a double flash. There's a double flash. And there should be one more double flash going off the side. And so this is this species signal. By the way, this is one of those that only flies for about 20 minutes, right after it gets sunset. So it's one of those that has a very brief flight window in the evening. And this is what he looks like, by the way. It looks very similar to a Phytinus paralis, but you'll notice that the yellow bands around his elytra are broader. And if you have them side by side, you'll notice he's also quite a bit smaller. And he's got a solid dark black bar going across his pronotum. So there are things you can see. This is another one. This is Photurus frontalis. This is one that some folks have started calling the snappy sink. I'll explain what that's about in a minute. You really have to look very carefully at this video because the flashes are very faint. But if you look at it carefully, you'll notice that there are multiple males flashing at the same time. So see if you can watch for that. And again, they're going to be faint. This is a species that flashes pretty much continuously while it's flying. And if you have high enough densities, the males start synchronizing their flashes and synchronizing the periods when they stop flashing. So again, the third species, a third different flash pattern. And we see this for basically all the species of fireflies. Now, one wrinkle is that a lot of Photurus have more plastic flashing behaviors. And yet another wrinkle is a lot of the timing of flashes can be influenced by the temperature. So they may flash slower when it's cool and faster when it's hot. Because like other insects, fireflies are what we call poikilotherms, which means they're essentially the same temperature as their environment. So there are variations. Some have different kinds of delays between male and signal and female response. Like I said, some Photuris have multiple flash patterns. Some do some kinds of other things with their light making ability, and probably the most notorious of those we'll talk about in a second. All right, so what's with the funny smell? When we talk about fireflies, I know everybody that's caught one has noticed that there's this kind of curious odor. Well, that may be related to the fact that a lot of these, particularly Photinus, produce steroidal chemicals that are called lucibufficins. And they produce these primarily to protect them from predators. They're toxic. They're really nasty tasting. I have a graduate student who on a dare put one in his mouth last year. So you can talk grad students into doing some weird things. And his lips were numb for about 45 minutes because of the lucibufficins that he picked up from that firefly. And so, as I said earlier, we think that they may have originally started producing light to warn predators at night that they're not something that they should be eating. There may be other defensive chemicals involved. As it turns out, lucibufficins are not terribly volatile, so they're probably not what we're smelling, but a degradation product of lucibufficins might be what we're smelling. So in any case, many fireflies are noxious, and they produce those chemicals to protect themselves. And if they have to, they'll reflexively bleed on their predators to help convince predators that they're nasty to eat. So here's a picture of a Photuris, and that bubble right here between its hind legs is a bubble of hemolymph that it's expressed to try and get me to let it go. And so that exception that we talked about when we talked about most adult fireflies don't feed, many Photuris females are aggressive predators as adults. And they're predators to a large extent on one particular kind of prey, and that's other fireflies. So in these Photuris species that are predaceous as adults on other fireflies, the virgin females watch for males of their species, and then when they see a male of their species, they signal like they're supposed to, like good girls, and then they mate. Once they mate, on subsequent nights, they watch for male Photinus, and some may prey on other species as well. And when they see a male Photinus, they mimic a female Photinus, and eventually that poor sucker ends up there. So this, by the way, is a picture of a Photuris eating a Photinus paralis under my back porch light I took several years ago. It still just tickles me I got the picture. Anyway, so they do this because Photuris harvest those noxious chemicals from the Photinus to protect their own reproductive efforts. And with those long legs, sometimes they'll actually catch them out of midair. They've also been known to steal dead fireflies from spider webs. By the way, lots of spiders don't like to eat fireflies, so fireflies will get in their webs and they'll go over there and cut them out. One other real quick phenomenon I wanted to mention to y'all is the fact that some fireflies do synchronize. This is probably the most famous of the synchronous species. This is Photinus carolinus, which is found throughout the Appalachians, actually, from certainly at least northern Georgia all the way up into Pennsylvania. And it's noteworthy because the males, when they're in their courtship flights, if the populations get high enough, synchronize their flashes. So this is a species that flashes about 8 times in sequence, and then they go dark for about 4 seconds. And what they synchronize is their dark periods. And so it's a pretty magical thing to witness. This is most well known at Elkmont in the Great Smoky Mountains National Park. But the more we know, or the more we learn, the more we know that that there are many other places where this species reaches densities high enough to allow them to synchronize in this fashion. And if you haven't seen it, it's something worth seeing. I just last summer discovered another population of synchronous fireflies at Grandfather Mountain in North Carolina. While I'm talking about synchronous fireflies, it's important to know that the more we learn, the more we know as well, that other species of fireflies synchronize. And so the one I showed you a little while ago, Photurus frontalis, in certain areas, for instance, at Congaree National Park in South Carolina, they reach high densities and they synchronize very, very precisely there. The ones in my backyard, I don't have densities high enough to get them to synchronize really well, but I'm pretty sure there are other places in North Carolina and South Carolina and perhaps other states across the Southeast where Photurus frontalis gets into high enough populations to put on a pretty spectacular synchronous show. And it is possible that there are other species that synchronize to some degree or another as well. And this is yet another thing that can be learned with folks like y'all participating in Firefly Watch. So with that, I'm going to stop talking, and if there are any new questions that I elicited, that'll be fine. Otherwise, we're going to turn the show over to Chris.
Yep, we just have one or two. And just to review, can you just remind us what the requirement is for the production of cool light, that chemical reaction?
So it's luciferin, the enzyme luciferase, oxygen, and energy source ATP. And we think, although this is still not terribly completely well understood, we think that one way they regulate the on and off switch is by regulating oxygen into the reaction. And by the way, we do something similar. If you've ever played with glow sticks or used a glow stick on a fishing rod or something like that, that's a synthetic version of this same kind of chemical reaction. But unlike fireflies, when the reaction it's spent, fireflies can recycle their luciferin and luciferase. And so they can continue to flash for their entire adult life.
Great, thank you. And so does synchronization improve reproductive success somehow? And do the females synchronize their responses too?
So we think that they synchronize so that everybody has a chance to look for the girls. And if they all at high densities, if they all just sort of randomly flashed, the females would never be able to sort out a male, and the males wouldn't really have much of an opportunity to see the females. In that particular species, the females tend to stay on the ground, and on any given night, there are far fewer females up and receptive and actually looking to mate than there are males flying around. So what you often see is the males will be flashing, they go dark and they're looking, and then they flash, and if they see a female respond, there may be 20 or 30 males will all roll down there trying to be the first guy there. And you get this ball of fireflies all fighting over a single female. But we think it gives them an opportunity to see, and also an opportunity for the females to actually tell what's going on.
Great. And does synchronization only occur in the evening?
Well, I mean, so Photinus carolinus is one of those species that doesn't really start flashing until it's flat dark, and they'll flash usually for about an hour to an hour and a half before they stop. So yeah, they only synchronize in the evening during their regular flash time, and they only synchronize if they're really high densities. If they're at moderate densities, the synchronization starts breaking down because they can see what's going on below them without the confusion, and the females can sort them out if they're not really high density.
Great. And do you need a special camera or lens to capture those night shots of the flashing?
Well, the camera that I use is a Canon SX 60HS. It's a regular kind of a super zoom point and shoot. You do have to monkey around with your settings, and you also have to have a steady hand, and you also have to be lucky, because if they're very far away from you, it's pretty difficult. There are camera rigs that are better for capturing fireflies, but frankly, I don't have the thousands of dollars I think it's going to take to do that.
Yeah.
But you can have pretty good success with some of the super zooms, and I've been pretty happy with my Canon. I'm not getting any kickback from any camera companies either, by the way.
Great, thank you. It was hard to tell in the video, but is the positioning during reproduction different than with synchronization?
So that picture of the Carolinas, that was a coupled pair. They were mating. So that might have been a little misleading. I actually had a video. Maybe we can see if it'll play. Yeah, doesn't want to do it. So I had a video that showed the synchronization, and with Photinus carolinus, it's not precise. The individual flashes aren't precisely synchronized. The dark periods are synchronized. And if you're looking out across, say, a wood, and this is a forest species that likes to be in the trees. If you're looking out across a forest, what you'll see is a wave of dark go across the hillside, followed by a wave of light. And it's really pretty impressive. Yeah.
Great. And it looks like, lastly, are there ways to attract more fireflies to your yard, such as limit light, certain plants?
So plants, probably not, except that it's good to not have invasive shrubs and stuff like honeysuckle and stuff like that, because it can interfere with their ability to see the females on the ground. Definitely limiting light is a good thing. Fireflies are attracted to light. I get pictures of them at my porch light because they come to the light. And so light can confuse their abilities to communicate effectively with other members of their species. But probably the most important thing is providing good habitat and good undisturbed habitat for the different species.
Great. And it looks like just one last question, and then we can hand it over to Chris. But are the female flash patterns similar to the males for the same species?
No, not necessarily. In fact, in Photinus pyralis that we talked about earlier, the Big Dipper. So the male has that roughly half-second long or so J-shaped flash. The female has a very brief little blip. So no, they're not the same. And so one of the challenges is associating. For one thing, it's usually much harder to find the females, because again, even on other species, on any given night, only some of the females are receptive, but all the males are on the hunt. So it's often much harder to find the females. But associating a female flash with the male flash pattern can be kind of interesting. I'll put it that way.
Great, thank you. It looks like that's it for questions at the moment. So thank you very much. And I think we can hand it over to Chris, I believe.
Okay. Do I need to do anything to get my screen out of his way?
Just hit pull up your toggle over the bottom of your screen again, and you'll get the screen share icon, and just hit that to stop sharing.
There it is. I see it. Oh, good. That's it then, right?
Yep. Perfect.
Thank you all for your attention.
Thank you, Clyde. We appreciate all that information. And I'm going to jump in now and share my screen. And let's see. All right, I'm going to pull up my PowerPoint. And I guess I'll just ask Robin, can you confirm that you can see that?
I can, yes, it's perfect.
Excellent. Okay. Well, thanks everybody. So we've had a really excellent overview of firefly biology and all kinds of interesting insights into the ways in which they interact. And what we're also trying to accomplish with this presentation is to certainly encourage you to get involved in Firefly Watch. And if you've already been involved in Firefly Watch, to encourage you to dive even deeper in some ways. And the first step in that process is to really encourage you to think about what you can do to try to identify which firefly genera you're looking at in your habitat, and potentially even describe flash patterns, maybe even from those flash patterns, make some some guesses about what species you're looking at. We've already looked at the morphology of these three major genera, the Phytinus, Phyturus, and Pyractomina, and we'll very briefly review that for you again, because as you participate in Firefly Watch, we would love it if you can make an effort to give us a sense of what you think you're looking at, what you think you're reporting on in your habitat, whether it's Phytinus, Phyturus, or Pyractomina. And then beyond that, to describe to us what the primary flash characteristics are that you're seeing in your habitat. So we'll talk a little bit about how we define flash characteristics so you can describe it more effectively. After that, we'll talk about the Firefly Watch observation procedure and a new opportunity that is kind of in its beta form right now, which is what we're calling Firefly Watch Pro. It involves a little bit more intensive observations for those of you who are interested in delving deeper. So I'll try to get to that as well in this presentation. So to begin with, what are the genera in your backyard? You've heard from Clyde what the possibilities are. Phytinus, Phyturus, and Pyractomina being the most likely ones, especially for those of us here on the East Coast. And you've heard a little bit about the identifying characteristics. So remember that this head shield is what we call the pronotum, and that in Pyractomina, there tend to be, in many cases, these dark margins along the pronotum. As Clyde described so well, the pronotum has a little bit more of a triangular shape to it. And the differences that we see in Phyturus, he's described as well, these wider legs. In some Phyturus, this is not true of all of them. In fact, if you looked carefully at the Phyturus frontalis image he had, you would not have seen those stripes, but this is a Phyturus versicolor. Some of the Phyturus, particularly Phyturus versicolor, tend to have these light stripes on their elytra. So those are some of the characteristics to try to bear in mind as you look at it. Just summarizing it, the darkened lateral margins of the pronotum in Pyractomina may be a helpful clue for looking at them. The pale stripes on the elytra of some Phyturus and the long legs and larger size of Phyturus in particular may help you to separate them out. We're also encouraging, particularly if you try out Firefly Watch Pro, to have folks send in pictures of the fireflies that they're seeing. And that can help us to look at them and see if we can make any judgments about what it is you're looking at. Now, I wish this information was a little bit more helpful, but Clyde has already warned you that finding females is going to be very difficult. There are some really distinct differences in the lanterns of the females of these three groups. Phytinus females, the lantern occupies only a small segment of one of their abdominal segments, a small portion of one of their abdominal segments. The Phyturus female, the lantern occupies just a portion of, sort of two and maybe a little bit of a third abdominal segment, but just not filling the entire one. And in Pyractomina, the lanterns occupy the margins of two segments. And that's in contrast, all three of these genera, the lanterns of the males occupy the full part of two segments as pictured here in Pyractomina, here in Phytinus, because this is a Phytinus being eaten by a Phyturus, another image of that. And you can see that this is a Phyturus female, because you can see that lantern is not quite filling the full segment. And it also gives you a good idea of what can be the rather extreme difference in size between Phyturus and Phytinus. It wasn't as striking a difference in the image you saw from Clyde, because he was showing you a picture of a Phyturus eating a Phytinus pyralis, which are amongst the largest of the Phytinus. This is a smaller Phytinus species being eaten by one of the largest of the Phyturus species, Phyturus versicolor. So there's a big difference. And then here's just an image of Phytinus male and female mating. The images that you've seen in Clyde's show after they have begun the mating process, they turn in that abdomen to abdomen position that you've seen. And as he mentioned, they stay in that position for sometimes up to 8 hours. But when they initiate mating, they do begin it head to head like this and stay in this position for a few minutes before turning into that other position. So a few other things, but again, just highlighting some of the differences. And the lanterns give us, if you can capture a female, which, as we say, is a little bit harder, gives us one other clue that you can look at if you're trying to determine what the genera are that you're looking at. Now, you've heard a lot about flash patterns, and if you figure out what genus you think you're looking at, flash patterns can really be, for most of us, the only way then to try to distinguish within that genus what species we're actually looking at. Because as you've already heard from Clyde, the species within a particular genus, they might vary in size slightly. We talked about pyralis here being particularly large Phytinus, but otherwise it's fairly hard to tell apart different species by any morphological characteristic. Really, the flash behavior is what sets them apart. So you've seen a video of the pyralis flashing about every 5.5 seconds or so, and having relatively long duration flashes. What this diagram can't show you is that sort of J shape as they swoop upward while they produce this long duration flash. But this is sort of the standard way we try to represent flash patterns. And if you go to the Firefly Watch website, you're going to see these same types of charts, and you can go in and use them as a reference to get perhaps, hopefully, some sense of what you're looking at if you're able to narrow it down to what genus you're looking at in the beginning. And so this also gives you the data about the female response delays. Please do remember what Clyde said, that the response delays and all of these flash patterns are temperature sensitive, so that as it gets colder, there'll be longer gaps between these male flashes. And as it gets colder, there'll be a longer delay before the female response. But if you're lucky enough, again, to watch a female response, or even perhaps to elicit one by using a pen light to produce a simulated male flash of your own, you can count off in your head. Sometimes that's easier than trying to use a stopwatch to get a sense of what the flash timing is that you're looking at. And I'm going to just quickly go through the three different genera in terms of their flash patterns. And actually, I'll just highlight again, one of the things that's striking about Phytinus is a very diverse group. They have flash patterns that range from these single pulses to pairs of pulses, like you saw the example of Consanguineus, and even trains of multiple pulses. Carolinus, that you were hearing about, that synchronized, produce these trains of multiple pulses with long gaps between them, but there's other species as well. And this is not an exhaustive diagram either. It's just our best efforts to represent some of the common Phytinus fireflies. This is Pyractomina, and Pyractomina tend not to have any of the double pulse flashes, at least in the ones that have been studied carefully. They tend to produce single pulses, sometimes shorter duration, sometimes longer duration, and trains of pulses, sometimes sort of flickering pulses at times. And that can give you a sense of what you're looking at there. You also heard from Clyde that in many cases, but not all, Pyractomina may have a more amber color to their flash, or a more orange color, and sometimes even a reddish color to their flash. So that's can be a potential distinguishing characteristic to look at when you're looking at those. And in this case, we've just done the diagram a little bit differently. Female response delay is just placed right next to the male flash signal as opposed to placed on a separate column. And then finally, here's a chart of Photuris. This one does not have any flash delays on it. Female flash response delays on it, but it shows you the variation. So frontalis, as Clyde alluded to, produces flashes very frequently, about every second. Photuris versicolor produces flashes in these long trains, and he mentioned that there's variability to their flash pattern. Versicolor probably has some of the most variability because sometimes they're producing these trains of 7 flashes, sometimes 6, 5, 4, and that can be particularly variable. But we can see many different kinds of flash patterns across the Photuris species. So in summary, flash patterns and sometimes colors can give you some sense, not only of which species you're looking at, but some sense of which of these genera you're looking at as well. And we're going to dive a little bit further into the individual flash characteristics, because once again, we're hoping that you will, whether you're doing Firefly Watch or whether you try out Firefly Watch Pro, that you'll try to describe to us something about the flash patterns you see. If you've done Firefly Watch before, you know sort of at the end of your entry for a particular night, we ask you what is the most common flash pattern you see. And so we want to make sure that you understand sort of the language that we use when we're describing these. So to do that, I've put up here a figure that shows both a single pulse species and a double pulse species. And so that's the first part of the terminology. Is when we talk about a pulse is our way of describing a single flash. And so if it's a single pulse species, it's producing one single flash and then repeating that at a regular interval that we call the flash pattern interval. We actually don't anywhere in either of the sort of versions of Firefly Watch ask you to report female response delay, but certainly you're welcome to. They are open response questions in the forms, and so you could give us more information if you wanted to. Female response delay is, of course, timed from the first male flash, how many seconds until that female produces her response. And then the other important piece of information is what we call pulse duration. And in Firefly Watch Pro, that's something that we ask about. Of course, it's very difficult to measure any kind of exact pulse duration, but we do ask you to tell us sort of, is it a really long pulse duration, more than a second? Is it a short pulse duration, less than a second? Give us a sense of that. Now, if you get to a double pulse species, then there's additional information you can report, because you've still got a pulse duration and you still got a flash pattern interval, how long between one, in this case, pair of flashes, starting from the first pulse of that pair to the first pulse of the next pair. But then there's also the between pulse interval. How much time is there that elapses between those two pulses? And that's information that can be helpful and informative in figuring out what kind of firefly species that you're looking at. And of course, although I've represented single pulse and double pulse species, there are those that can produce multiple pulses within a flash pattern, and that's important to report as well. So just to make sure that we understand what those different characteristics are, we'll jump back in to these flash patterns again. And we'll look, for instance, at Pyractomina borealis. And Clyde showed us some great videos. In this case, the Firefly Watch folks have put together some nice animations too that just give you a sense of what we're looking at. So Pyractomina borealis produces flashes. And by the way, as Clyde mentioned, these are species that come out relatively early in the season. So for instance, Massachusetts, Pyractomina borealis is already out, whereas many of the other Photinus species have not come out yet. Now, I'm sure in other warmer places they're out, but here the Photinus are probably just, many of the Photinus species are just going to be coming out in the next few days, but borealis has been out already. So you can see here a flash. This is a single pulse species with a flash pattern interval of a little over 2 seconds. And so if we watch this, borealis are also a species that flies in and around trees, often on the margins of forests, but staying close to the trees. And here's an animation just showing that flash pattern. And we're using it to sort of get you to think about this. You can imagine yourself being out in the field and watching a flash and then beginning to count. Flash 1 Mississippi, 2 Mississippi, 3, okay, 1 Mississippi, 2 Mississippi. Can't quite get to 3 Mississippi in our counts. So we say, all right, this seems to have an inter-flash interval, a flash pattern interval of something between 2 and 3 seconds, so about 2.5 seconds. So that's the kind of thing that we're hoping people will take a look at. And as we said, Pyractomina males tend to produce those single pulses or flickering flashes, and they can be distinguished by looking at how long the duration is of the flash, and that those were, as you saw, fairly quick flashes, and those flash pattern intervals. In that case, an interval of about 2.5 seconds or so. As we come back to looking at Photinus, I'll move from Pyralis, which we've already seen, to Photinus ignitus. This is, as we mentioned, if you're in Massachusetts, and by my count, roughly half of you were in Massachusetts in the group tonight, you will not be able to see Pyralis, most likely, unless there's been some major shifts. We certainly encourage you to look and tell us. That's one of the things we're looking for people to report on in Firefly Watch Pro is there any evidence of an expansion of Pyralis's range further north? But Photinus ignitus, like Pyralis, is another single pulse species with pulses repeated about every 5 seconds and a very long female response delay of about 4 seconds. They don't have as long flash durations as Pyralis. And so again, if we look at an example, we can imagine ourselves out in the field. They fly, they are a grassland species flying mostly over fields, and we can go out and watch them and count in our head from a flash, 1 Mississippi, 2 Mississippi, 3 Mississippi, 4 Mississippi, 5 Mississippi, 6 Mississippi. So around 5.5 seconds. And again, you can do this with a stopwatch as well on your phone, or try to do it the counting as I have. But again, hopefully you can use these kinds of techniques to give you a sense of what you're looking at. And you'll know that if it's about 5.5 seconds, then it's more likely ignitus, than it is sabulosis. And we've got it listed here at about 5 seconds. Do remember that it's temperature dependent, so it's going to be imperfect, but please report back to us on what you see. We would love to know and to get an idea of what's out there. I also can highlight the double flash species, Photinus greeni. You've already seen an example of a double flash species, Photinus consanguineus, that has a fairly short interpulse interval. Photinus greeni, on the other hand, has a longer interpulse interval. There's the first flash, second flash, and then the interflash interval until the next one. So we can get an interpulse interval, 1 Mississippi, 2 Mississippi. Okay, about a 2-second interpulse interval. We can also try to get the interflash interval. There's the first flash, 1 Mississippi, 2 Mississippi, 3 Mississippi, 4 Mississippi, 5 Mississippi, 6 Mississippi, 7 Mississippi, 8 Mississippi. Okay, maybe about an 8-second interval between those flash patterns. So those are some of the things that we ask you to look for. Photinus males do produce these single, double, or in some cases, like Photinus carolinus, multiple pulse flash patterns. And you can use that along with, as we said, female response delays to identify those species with some level of accuracy. Certainly, we want you to give it a try, report back to us what you're seeing. And then finally, as we mentioned, the Photurus. We'll just show you, again, another animation of what a Photurus versicolor might look like to you, producing these trains of multiple pulses. You're going to see them in a lot of places. They're flying all over. They're very active, flying in the trees, but also over the fields because they are, especially the females, predatory on the Photinus. And so there's just an example of what that Photurus versicolor might look like. So the Photurus produce those flashes. Please do remember, though, that Photurus also mimic Photinus flashes. So I can't tell you how many times I've been out there, especially if I'm looking for a Photinus female and I'm out there with a pen light trying to elicit female responses as if I'm a male firefly. And I go zoom in, turn on my headlamp. Oh, what have I found? And in fact, I found a Photurus instead of a Photinus that tricked me just the way it was trying to trick Photinus males. So with that, I will now pause for any questions that you might have.
Great. Let's see. I am in Minnesota. Do you have any data as to the types of species in Minnesota, or do you think it would be similar to Massachusetts?
So Minnesota, I can't say off the top of my head. There may, again, if you go to the Firefly Watch website, I think we do have a few range maps of some of these species up there that are drawn from the work of James Lloyd. And this is work he did back in the 1970s, which is one of the reasons we really encourage people to report back what they're seeing, because we feel like it's very possible that things have changed since the 1970s. I do think Minnesota is a place where, if I'm recalling some of the things I've heard about and seen, it may be that Pyralis has made it out to Minnesota. So there's going to be, I don't want to hazard any guesses. I haven't watched it firsthand myself, but I wouldn't be surprised to see any number of the Phytinus and Pyralis, the Phytinus and Pyractomina and Phytura species we've seen. I wouldn't rule out, any of them necessarily for Minnesota off the top of my head. So I should give Clyde a chance to respond to that too, because he may have some insights as well.
Well, there are some species that are kind of more Midwestern, and so the diversity in Minnesota is probably not going to be greater, but it's probably going to be a little bit different in terms of species composition from, say, Massachusetts.
Yeah, that's a good point. One of the multiple pulse species, Phytinus consimilis, is very much a Midwestern species. And so that's something, again, we don't find here in Massachusetts, and I don't think we find it too much on the East Coast in general, but you will find it out in the Midwest.
Great, thank you. And there was one comment that the animation was very helpful, so thank you for that.
Good.
All right. In Massachusetts, is there a certain time in the summer when you typically see the most fireflies? Any way to predict a good firefly night in advance?
Yes. So, again, in terms of Firefly Watch, we encourage people to be out early and often, because, as Clyde alluded to, there are species that come out for short periods of time early in the season, some species that come out later. And as he also mentioned, it's different times of night. So you've got to be aware of looking at different times of night. With that said, you can pretty reliably in Massachusetts say if you go out around the 4th of July, preferably not on the 4th of July, because you're going to be blinded by a lot of fireworks that are going to completely throw off any efforts to identify flash patterns. But those that early period of July is really when things peak for many firefly species, but again, not all of them. But if you were looking for a particular time, that's what I would point to. In Massachusetts, they are virtually all gone by the beginning of August. So please do not wait. Get out early and often, and we would love to hear what you're seeing and get those reports.
Great. It seems that it would be challenging to discern flash patterns when population is high. Any advice? Also, what is the range for female detection of males?
So the answer to the first question is that's absolutely right. And some fields are going to have multiple species active over them, and at those periods of time in early July, it may be very, very difficult to do the kind of flash pattern recognition that we're talking about here. And again, that just reinforces the importance of doing this over a long time span at a particular site, because when males of one species are first emerging, they may not overlap as much with the other species, and you may be able to have those time periods when you can more easily identify them. It can also become easier as it gets cooler. Now, remember, you'll have to take into account the cooler temperatures when you're trying to figure out what species it is, because something that had a 5.5 second delay might go up to a 7-8 second delay, or even inter flash pattern interval. But but that can make it easier. So I don't have a simple solution to that. We just encourage you to, to do your best and tell us, what you think you're seeing. You do get better at it the more you practice. There's no doubt about that. And learning to follow, to try to track a single firefly, even if there is a bunch of other fireflies active around it, trying to sort of track the flight pattern of a single firefly and see its flash pattern. That's just an acquired skill that comes with being out there a lot and watching them. So I'm not going to say it's easy, but I'm going to encourage everybody to give it a try.
Great. Is there certain, oops, go ahead.
Yes, when they get to those really high densities, just sit back and enjoy the show.
Yeah.
Excellent advice. Thank you. Is there a certain type of light we should be using?
So if you're talking about, I guess there's a couple parts to that question, because one of the things that you may want to do if you're getting really enthusiastic about this is to have a headlamp that you've actually put a colored filter over, because that allows you to ensure that you're less likely to disturb their behavior. And as you heard from Clyde, although for many insects we use red filters because they don't see red light, for fireflies that doesn't seem to be as reliable. So in my lab, we've always used blue filters instead. With that said, it's best to just keep your headlamp off as much as you can anyway. And but that may not be the key to the question. If you're thinking about my idea of using a pen light to simulate flashes, there are, in that case, it appears that for many of these species, even though there can be differences in the color of light they produce, they don't seem to be that sensitive to the colors of light that we produce, especially if we're just producing a very white light as a stimulus flash. So in that sense, I often will just use an off-the-rack pen light. There's also a Firefly fishing rod design that I think we have up on the Firefly Watch website, where you can use an LED, and a battery and an old fishing rod to be able to hold that and flash further away from you. So I think that's still up there. So please, check it out.
Great. Let's see. How likely is it for you to witness only one species at a time?
It really depends where you are. And so I don't have the good fortune of Clyde where I have so many species on my property. My property is not quite as big either, but I only have one species on my property here in Massachusetts. On the other hand, I've gone and visited, Audubon properties around central and eastern Mass. And, it just varies so much. There's some that will have these incredible meadows with, 5-6 species active at a time, just over even in the same area. And then again, there's others that, where there's very discrete populations. Some of my research was done here in Concord at the Old North Bridge, something that folks in Massachusetts may know as a landmark, or even some of you have traveled to Mass on historical tours. And, there's one, just a very distinct population on a hillside there that's just Phytinus marginellus, and it doesn't get confused with other species, which sometimes makes it easier to observe. So it does happen for sure.
Great. Could you please repeat the species often found in the Midwest?
Well, I guess I didn't mean to suggest they were often found there. Just to point out that, to my recollection, Phytinus consimilus is a species that is in the Midwest, but is not found on the East Coast. And on the other hand, we've talked a bit about Phytinus pyralis that's not found in Massachusetts, but is found further south from here and does seem to be pretty widespread even into the Midwest. So again, it varies from species to species, but Phytinus consimilus is one that I recall being found in the Midwest, but not found on the East Coast, certainly not found in Massachusetts.
Great. And I'm a teacher in the St. Louis, Missouri area. Are there any local researchers that you know of in this area? I would love to build a summer enrichment program for my students and others.
Nobody comes to mind for me right now, but please don't hesitate to follow up with us on Firefly Watch. It may have been you, it may have been someone else who I know sent an email to Firefly Watch recently about that, and I know we're sort of reaching out to the different people in our community to see if we can find anybody that we've heard of or know about. Yeah, don't hesitate to ask us. I don't know off the top of my head.
Great. And are we finding predatory changes from the Photurus females, and are male Photinus getting smart and not getting eaten by Photurus females?
Well, obviously, this is something that that we imagine happening over long periods of evolutionary time. I alluded to before the idea that some of these species of diurnal fireflies that are active during the day, don't have lanterns, that we think that they secondarily lost bioluminescence. In other words, they evolved from species that had adult nighttime bioluminescence, we believe, just based on the evolutionary patterns. And we think one of the reasons for that may be that they were driven into this new evolutionary niche because of the predation going on by Photurus fireflies at night. Of course, it's a hypothesis. It's something we have to explore more to test, but it's one interesting possibility to explain why there are some daytime fireflies and why they seem to occur within a phylogenetic tree, within an evolutionary tree in which the vast majority of the other closely related species to them do flash at night. And so that's one possibility. We also, when we look at the incredible diversity of Photinus firefly flash patterns, we also wonder if it's possible that some of that diversity is driven by Photurus predation. In other words, if we know the Photurus evolved to mimic one particular female response pattern, wouldn't that favor the evolution of changes in the female response pattern that might then cause new species to arise with new female response patterns that the Photurus has yet to evolve? So we imagine, yes, this kind of evolutionary arms race going on over thousands and millions of years, but obviously not something that we think we can see in our lifespans necessarily.
Yeah, I'd add to that a lot of these things, they can happen rapidly, and then there may not be change for a long time. So if you think about the recent history of North America, most of the area that we're talking about was under ice 18,000 years ago. And so a lot of these relationships, a lot of these distributional patterns and all only reestablished, over the last several thousands of years, which seems like a long time, but in evolutionary terms is not really that long.
Yeah, that's a good point. Now, Robin, can I ask that would it be possible to hold the rest of the questions now?
We certainly can. Yes, I was just looking at the time.
Yeah, I want to be sensitive to people's time so that we can wrap up. There's not many more slides that I have, but we can wrap up just talking a little bit about the Firefly Watch and Firefly Watch Pro process. Then I'll keep answering these questions, and Clyde and I will keep answering these questions afterwards. And I don't mind going over time doing that, but I just know if other people had other things planned for their evening, I'd love for them to hear about what we're doing with Firefly Watch.
Perfect. Thank you.
Okay, great. So as we continue to talk about this, you've already heard me plugging this quite a bit. And again, many of you may already have been involved because that's how you found out about this talk tonight. But if you haven't, please, we encourage you to visit our Firefly Watch Citizen Science Project. Oops, that was not meant to change quite so fast, because it's got a variety of things, information on how to participate, how to submit observations. It's got a number of frequently asked questions, some information about myself and other researchers involved, the opportunity to go in and view and explore our data, and also a number of resources. And that's, first about fireflies, which will include some of these flash pattern charts we've talked about, but other resources about fireflies as well. So please do visit that to learn even more. Please do consider joining, if you haven't already, the Firefly Watch. Identify a firefly habitat to study, even just your own backyard. Register online at the Firefly Watch website. And the process involves sorry, it's switching a little faster than I wanted to, but the process involves describing your habitat online. And what we ask for in the standard Firefly Watch process is to report on your habitat about once a week. So take the time once a week to go out. We have an online data entry process, but there's also a PDF of the data entry information. So if you want to do it by hand and then go in later and do it online, note the date, the time, weather data, and yeah, see, it keeps, I must have put in an automatic change there, but what we're looking for in Firefly Watch is for 3 10-second intervals, count just the total number of flashes that you see. It's giving us a sense of the overall density of fireflies, the kinds of things that we talked about, when they're out, how early they're coming out. All of that is interesting to us. Put it into your Firefly Watch system. And again, many of you may have been doing that already. And I should also note that on the left-hand side here are just images of some of these daytime fireflies to show they look very similar to the nighttime fireflies. You've already seen a picture of one, but this is just a line drawing of Lucidota, one of the genera of daytime fireflies, and the other one was Alichnia, another of the genera of daytime fireflies. And we see both of these in Massachusetts and elsewhere. They're pretty widespread species. Now, the last thing I really want to tell you about is that if you've been doing Firefly Watch in the past, we want to encourage you to think about expanding your activity. Try this new pilot project we're doing called Firefly Watch Pro. Firefly Watch, as I mentioned, asks you to go out 1 time a week. In Firefly Watch Pro, we're asking you to go out 2 times a week. In both cases, you're providing some location coordinates. But in Firefly Watch Pro, we're also going to ask you to be really careful about exactly where you're looking on each night. It's certainly a great idea to do the same thing if you're in Firefly Watch, but to try to help ensure that, we ask you to report in Firefly Watch Pro the cardinal direction, the direction you're facing as you do your observation, to come back and look at that at the same time. And we're asking you also to even estimate the overall area in square feet that you're looking at, so that we can get a sense of what the firefly densities might be. We can compare your data to someone else's data potentially. At least we're trying to see how well that works. Obviously, in Firefly Watch, we're always asking you to provide habitat type, whether it's been mowed, information about the weather, information about any artificial light at the site. For Firefly Watch Pro, we've expanded some of those questions there. Not just whether there's mowing, but how frequently, not just whether there's artificial light and how far away it is, but the number and type of light sources. We're also asking for information about pesticide use, whether there are fire ants on the site, and a number of other disturbances that might influence the site. So we're looking for some additional data. And then, as I mentioned before, for Firefly Watch, we ask you to go out and for these 3 10-second intervals, which can, all be at the same time, over a period of just, a half hour or so, or even less, 10 minutes or so, you might take those 3 observations and just tell us the overall number, first total number of flashes you've seen. And then we also ask you to tell us the overall number of flash patterns. And one thing I didn't put on this slide is we do ask you in Firefly Watch to tell us what seemed to be the most frequent flash pattern you saw. And that's why I spent so much time talking about flash patterns, so you could use some of the same language we do when reporting on those flash patterns. But when we get to Firefly Watch Pro, we're again asking you to do those 3 times for 10 seconds, but we're actually asking you to spread them out over the night. And instead of just counting the total number of flashes, each of those 3 different time points over the night tell us the number of flash patterns you see at each of those time periods. And also, if you can, and we already talked about how difficult this can be, but if you can identify one particular species and look for just that flash pattern to give us the number of flashes you see of that species in those 3 different time periods. And again, you may not be able to say it's definitely this species, but you're saying to us it's definitely a species with this flash pattern. And the one other thing that doesn't show up on this slide is that we ask you in Firefly Watch Pro, and certainly we welcome you to do the same with Firefly Watch, but we don't have necessarily the same mechanism to do it. But we ask you to submit a picture of, if you can, capture this firefly that you think you've identified by its flash pattern and send us a picture of it. So that's our goal, to try something even deeper, get more data. And for those purposes, we're using an online system called Anecdata. And so that's the general format for entering your data is pictured here. This is how you provide your location data, the cardinal direction that you're looking, the size of the focal area in square feet you're looking at. You give us your email because we're going to be, trying to work with you on this. And then there's 4 different data sets we're looking for. There is the one I already alluded to, a number of questions about the habitat. There's the one I mentioned about how is firefly diversity changing over time? So in other words, over the course of your night and then over the course of multiple nights, how are the number of flash patterns changing? And then data set 3 and 4. 3 is a really, really special little question. I've kind of alluded to it already. We're wondering about this really widespread firefly species, Phytinus pyralis. Is it entering new areas? So we just want you to look for that in particular, and when you saw it. And then finally, here's the individual species observation data, and the opportunity at the bottom, Anecdata lets you upload a photo. We're also asking that folks who participate in Firefly Watch Pro also upload their photos to iNaturalist. It's another great resource for looking at these things. Now, I know that we're right at the 9 o'clock time, so I want to turn it over to questions. So these next slides, you'll watch me sort of fly through them because they're just to show you that one other thing about both iNaturalist and Anecdata, unlike the data entry for the standard Firefly Watch, this Firefly Watch Pro data entry all has a mobile app-based interface. So you can add your observations in Anecdata on your phone, and you can also upload to iNaturalist on your phone. And now we'll turn it back over to questions.
All right. Let me get back up to where we were. Sorry, give me 1 second. Lots of stuff has come through, and then people. All right. Is UV light the same as blue light?
Well, no, in the sense that UV light would not really help. Much of the UV light would not help you to see as well. I mean, it would help you a little bit, I guess. But and again, there are many insects that are sensitive to ultraviolet light. So, you might be with UV light attracting a lot of daytime insects that you didn't want to as well. So I guess I would be cautious about using UV light. What we do is we just, again, we have headlamps, and we, purchase, from, sort of science education companies. Often will sell you packets of different filters of different colors, and we just, retrofit them, modify them, tape them onto our headlamps. So that's our quick solution for that. But as I said, it's not necessary either, because the truth is even blue light can be, in my experience, can be disturbing to firefly behavior. It can be hard to observe it, and if you're trying to, it's worth a try. But in terms of catching fireflies, trying to identify them, I often just keep my headlamp off, keep my headlamp off, keep my headlamp off until I think I've got one in my net, and then I'll turn on the headlamp, and it won't matter to me if it's got a blue filter over it, just white light, because at that point I know I'm going to be disrupting the behavior anyway.
And you're using the filter more to protect your night vision than you are anything else.
Yeah,
that's the real key to that.
Great. Where do the females usually lay their eggs?
So females, and it's going to vary by species. Most of my work has been done on grassland species, like Phytinus pyralis, Phytinus ignitus, and Phytinus greenii. In those cases, females lay their eggs, in the soil at the base of a plant. As you heard Clyde describe, females are not particularly active. So in my research, I was able to, for instance, capture a female at a particular site, mark her with a dot of paint, put her back on her perch, mark it with a flag in the soil, and go back night after night, and she would still be in that same spot night after night. And presumably, when she came to lay her eggs, she would just be crawling down to the base of that plant she was on, or flying down very gently, because these species I'm talking about, the females do have wings that I don't think they can fly very well with, but and laying their eggs right there. But obviously, there are other species with that may be laying their eggs in other areas depending on the habitat they live in, and Clyde can probably expand on that some.
Yeah, so and for a lot of the tourists, for instance, as females are so active, they can disperse quite a ways from where they started out before they finally settle to lay their eggs. But most of them are going to lay them in the soil or under leaf litter. The woodland species, a lot of them will be laying it right there at the interface between the soil and the leaf litter.
Great.
There's one other thing that we should point out, just for the purposes of people's general knowledge. We're talking about North American fireflies here. There are a number of Asian firefly species with aquatic larvae. And so if you're ever traveling to Asia, have an opportunity to see those fireflies, it's good to know that actually their larvae live in very different habitats than, in some cases, than the North American larvae, which ours are all terrestrial larvae living in soil and leaf litter.
Great. And just for some clarification, do we count the total number of flashes or the number of fireflies? The data entry form says fireflies and not flashes, so just want to be sure.
Yeah, so what we've been asking for the standard Firefly watch, even though I was noticing that today as I was looking over it, what we really mean is the total number of flashes, because we don't think someone can distinguish the number of fireflies, because, again, the same firefly will flash multiple times. And so as long as everybody is just counting the total number of flashes, we're getting at least some rough estimate of what the population is like in that site. Now, you can imagine there's some imprecision to it, which is why we're trying out this Firefly Watch Pro, to try to get slightly more precise data by asking the questions in a little bit more careful way. For instance, counting the number of flash patterns observed of a particular species and things like that. So, but yeah, for standard Firefly Watch, just the total number of flashes you see in that 10 seconds.
Great. And in Firefly Watch Pro, what do you mean by different times of night?
So obviously the challenge there is that it varies from habitat to habitat what's going to be useful for you, depending on what species you're looking at, because you heard Clyde describe, some that are out for just, 20 minutes at a time. So, you're welcome to adjust it based on the habitat you see, but we encourage people when they first go out to observe about every hour from dusk, so that, if it's getting dark around 8 and then you go out at 9 and then you go out at 10, we feel like that's the best way to capture the full diversity of fireflies you see there. Of course, if you do that for a couple weeks, or 2-3 weeks, and a couple nights a week for that, and you're just, I'm sticking around here till 10 and there's never anything here anymore. We're not saying you have to absolutely stick to that, and especially if it means you can get more observations of an individual population that you're looking at that it turns out is only there for a half hour or so, or 1 hour of that time period. So there's some flexibility there. You're going to be reporting the times of those observations anyway, so we'll know what it is in the data. But we just don't want to, because some fireflies fly very early in the evening. Photinus pyralis is one of those species. Around here, Photinus greeni is one of those dusk flying species. We don't want you to miss those, and because some come out much later at night, we don't want you to miss those as well when you're first assessing a particular habitat.
Great, thank you. All right, I'm going to do a last call for questions, because I think that was the last one that I have in chat. All right. So I think that's it for questions. So I want to thank both Chris and Clyde so much for your awesome presentations, so much great information. And thank you all for choosing to spend your evening with us here at Mass Audubon. We will be following up post-training to ask for feedback. So please, please, please fill out those feedback forms. We're always looking for ways to improve our offerings. And then we will also share with you some information on how you can further support learning experiences such as this and the critical research of our supporting scientists through donations to Firefly Watch. And moving forward, if you have any questions, please, please, please don't hesitate to reach out to us directly at fireflywatch@massaudubon.org. And thank you again for joining us this evening, and go forth, find fireflies, report them to the Firefly Watch database, and be well. Thank you again, everyone.
Robin, can I add one more thing in there?
Absolutely, please do.
That the email that you were just given, if you are interested in participating in the Firefly Watch Pro project, this was mentioned in the publicity for this, but we ask that you email that same email address, but put in the subject line Firefly Watch Pro, and we'll be developing a separate mailing list for that particular population of people who want to try to do that. We can give you support in using Anecdata and get you up to speed on it. So that's just one other note. Please, if you're interested in that, email us about that specifically with Firefly Watch Pro in the subject line of the email.
Wonderful. Thank you so much. All right. Thank you, everyone. Have a great evening. Be safe and be well. And we look forward to hearing about all you are finding out in the field through the data that you upload. Thank you.
Thank you for organizing this and for moderating it, Robin. So it's great.
My pleasure. I learned so much. It was great.
Thanks for the opportunity to participate. Take care.
Bye, everyone.
And have a great night. Thank you.